Okay, this is a relatively short screencast on meiosis 1, the first part of meiosis. Let's just take a quick review of meiosis. So this way we don't get all the slides up there. All right. Remember, we are producing sex cells here. We are producing gametes, sperm and egg cells. The main portion of the idea of meiosis is right here, to reduce the number of chromosomes by half, the two end number to the end number. A diploid cell, which has the full number of chromosomes, to a haploid cell. Now, I'm just covering here a germ cell, a cell that is going to develop into a sex cell may enter into have a short interphase where it prepares for division. DNA replicates as it has to. The cell increases in the number of organelles, especially mitochondria, just like mitosis. All right, you're going to stop here and you're going to listen to the animation that's attached to this, which will take you through this. And then I'm going to run through the important points fairly quickly. All right, because it's pretty easy. All right, so you just listen to the screencast, which is a good thing. All right, so let's go through prophase. Now, the big changes, the big differences between mitosis and meiosis happen right here. Anything, you want to look for them, it's mostly similar, but right here, all right, prophase 1. Prophase 1, you have all the notes, excuse me, called, all right, crossing over that occurs. All right, so first of all, take a look at the homologous chromosome. The homologous chromosome pair up and form what is called a tetrad, 4-chromatid, a tetrad. And it happens, it's called a synapsis. These are just words, boys and girls. A synapsis, not a synapse. Okay, so the homologous chromosomes. And you'll notice that the DNA gets so close to each other that they seem to overlap in certain spots. The spot is called a chiasmata, and that is what is called crossing over. When crossing over occurs, DNA is exchanged, not in all, but in certain segments to increase further diversity. So in the next slide here, when we bring up the metaphase slide, you're going to see a blue chromosome with a little red segment and a red chromosome with a little blue segment. Same thing going on with this one because DNA has further exchanged. The big deal of metaphase 1, we still line up, but the whole homologous chromosome is going to line up with its pair, not single file like in mitosis and meiosis 2. All right? Anaphase 1, pulling apart because that's what anaphase does. Except here, the whole homologous chromosome separates and moves to the pole. Unlike mitosis, where the homologous chromosome separates, here, the entire homolog goes over. All right. If this happens normally, which it usually does, it's called a disjunction. A disjunction. All right. The sister chromatids attached and remain attached to it. Telophase 1, you have what happens in telophase. You have a temporary nuclear membrane forming around it, around the chromosomes that are separated into two new cells. Cleavage furrow, remember telophase, and cytokinesis happens. From here on in, these cells are haploid cells because we're beginning to half the number of individual chromosomes. Cytokinesis occurs, all right, simultaneously with telophase. 2 daughter cells in a plant cell plate. In animal cell, a cleavage furrow. However, these cells at the end of cytokinesis 1 are not sex cells. They enter a short period of interkinesis, followed by meiosis 2. Let's take a look at the picture then. All right. Here, germ cell. Chromosomes have duplicated. Prophase 1. All right, prophase 1. We have lining up metaphase. We have anaphase pull apart. We have 2 new cells and 2 new cells ready to go into the next phase. So notice here, 1, 2, 3, 4, a diploid number, 1, 2 in this, a haploid number. All right, so we review meiosis 1. We'll be ready for then meiosis 2.